Hello everyone, welcome back to my channel Better to Classroom. Today's our topic is the continuation of our last video. A reminder, our topic was about the conjugation. Conjugation is a type of horizontal type of genetic transfer where two bacterial cells are placed in contact with each other, and with the help of your conjugation tube or a sex pilus, the genetic material is transferred from one bacterium cell to another bacterial cell. So there were, as you have seen, mainly three types of conjugation types. First one was your F plus to F minus conjugation. Second is your Hfr to F minus conjugation, and third is your F prime to F minus conjugation. F plus stands for your fertility factor, which also acts as a donor, which means your F plasmid is present in this bacterial cell, whereas F minus stands for absence of an F plasmid in a bacterial cells, which also act as a recipient cell. So the topic is about our Hfr to F minus conjugation. What does your Hfr stands for? Your Hfr stands for high frequency of recombination. It is rarely seen in E. coli strain. And your F minus stands for your bacterial cell having no F plasmid. So what is Hfr? And Hfr strain is a type of bacterial cell. It's a type of bacterial strain where F plasmid is incorporated within the main genetic material. So let me make a picture for your better understanding. Here is your bacterial cell. This is your F plasmid, which is a double-stranded, and this is your main genetic material. So this is your F plus, and let me make another bacterial cell. This is your another bacterial cell. So an Hfr means your this is your main genome. This is your main genome together with the integration of your F plasmid. This is your main genome, and this is your plasmid. Clear? So this is what is in your Hfr. So how this process starts, let me explain it. So let's start. Take two bacterial cell. Let's make two bacterial cell. This is your F plus, which means there is an F plasmid with the main genetic material. This is F minus, which do not have your F plasmid factor, but only the genomic material. When both the bacteria come in contact with each other, the sex pilus, what they does is they form a tube known as conjugation tube. They form a tube known as a conjugation tube. This conjugation tube is what helps both the bacteria in transferring the genetic material. So let me make another bacterial cell. This is your plasmid, and this is your main genome. This is your conjugation tube. This is the main genetic material. So what happens during the process of conjugation? The plasmid is double stranded. Here at the portion of your ORIT region, one strand, with the help of enzyme known as relaxase, it cuts one strand of double stranded DNA and is transferred along the conjugation tube to the another bacterial cell. But here what happens? For the process of conjugation, or for the transfer of your bacterial cell, the time required is minimum is 100 minutes. The time required for the transfer of genetic material to happen completely, it takes minimum at least 100 minutes. So here what happens is, before the completion of your 100 minutes, your conjugation tube, it breaks off or is disrupted. Why? Because it's made of your cytoplasmic membrane, and it happens to be a very fragile structure. So due to this reason, or it can be any other reason, so what happens, any other reasons like environmental stress, so what happens when this conjugation tube stops or disrupted, so obviously there won't be complete transformation of your plasmid, your F plasmid. So here what happens is, here is your main genome. This is your single stranded DNA, but since plasmids are capable of replicating independently, therefore another strand of your DNA is replicated as the other strand, as soon as the other strand is transferred. So what happened? Your conjugation tube broke down. So here the genome of your other bacterial cell will somehow look like this. This is the main genetic material, and this is your plasmid. So here, as you can see, that there was no complete transfer of plasmid. Why? Because the disruption of your conjugation tubes, the minimum time required for the conjugation tube is 100 minutes, but due to various reasons, what happens is the conjugation tube that disrupts or it breaks down, and because of that, the whole of your F plasmid is not transferred. So what happens is your only a portion of your F plasmid is transferred. So this is how the genome of the other bacterial strain will look like. So this is what happens. So afterward what happens is the fate of this incomplete plasmid is it can possibly happen in two ways. First, what it can do is, because of its incompletion, it tends to degrade. One possibility is that it will degrade because of its incomplete transfer. Another one is it recombines with the main genome, with the main genome, which is your this genome. So this is how your final bacterial genome of your final bacterial cell would look like. So either it will degrade, or either it will recombine with the main genome. So this is your main genomic material, and this one is your plasmid part. So this is what your Hfr strain is. Clear with the process? Let me focus it so you can see it well. So this is what is your Hfr strain or Hfr cell of bacteria. Hfr stands for your high frequency of recombination. Why it is called high frequency of recombination? Because this strain is most capable, or we can say it has high frequency. It has high frequency of transferring genetic material, to another bacterial cell. Why bacterial cell? Why? Because it has high frequency of transferring genetic material to another bacterial cell. As you can see here, in the main genome, there is also the integration of your plasmid. So we can say this whole genome is quite unstable. So what happens is, what did we say? We said it. We can say that it is quite unstable. So when it comes in contact with another bacterium, which is your F- bacterium, it has high probability, or it has high tendency of transferring your genetic material to your other F- bacterium. So this is your Hfr cell. This is your Hfr cell. This is your Hfr cell, and this is your another bacteria. So in case when your Hfr strain, they act as your donor. So when they act as a donor, it has high capacity of transferring your genetic material to the other bacterial cell, or you can also say it has high capacity of being a donor. So this is the reason why it is termed as high frequency of recombination, and it is mainly rarely seen in E. coli strain. So this is the whole topic. So if you have understood it, please don't forget to like, subscribe, and if possible, please share this to your other students or like-minded people, so it will be helpful in growing my channel. So thank you.